There we go. All right. Let's see who's going to come on in today. I'm going to give it a moment. Let everybody come on in. It is Wednesday, which means it's Teen Science Cafe. Let me get my things open here. Chat. All right. So welcome, everyone. For those of you who have never been here and have not met me, I am Lauren Traster. I am the 4-H Teen and Leadership Program Coordinator, and I coordinate these weekly science cafes. So welcome. I see some of you know the drill. Hi, Lily and Simon and Jack. So what we like to do is first start out by just opening up the chat box and introducing yourselves. I'm also going to pop into the chat box. If you need our closed captioning, there's a link that you click on and you will be able to follow the closed captioning. So I see Sage is back with us from Maine. Amelia's here from Greensboro, and Knight's back. Knight, if I remember, you're from Derby, maybe. I know you're up in Orleans County somewhere. So the rest of you, find the chat box, open it up, and introduce yourselves. Hi, Elizabeth from Pennsylvania. I should let our presenter know, even though this is the Vermont Teen Science Cafe, we do get participants from all over the country, as we see from Sage and Elizabeth, and I bet there'll be a few others. So Theo's here from Vermont. Hi, Gabby. It's okay. Welcome. I'm glad you could join us. I was telling our presenter that we sometimes do get participants in younger grades, and that's okay. Do your best to follow along, and you can always ask questions. So Fuzzy's back with us, and Wisteria and Cosimo are with us today. Fantastic. Who else do we have with us today? Well, continue to introduce yourselves. Oh, Matthew's from California. Nice. I love it when we get both coasts. Well, we're not really the coast in Vermont, but we're coast adjacent. So keep introducing yourselves in the chat box, because it's always so fun to just see who's here and where everyone's from. But I'm going to continue on and just remind everyone what our protocols are here when we're in Zoom. So you all are muted. The only people who have access to the mic are myself and the presenter, but it does not mean that we want you to stay muted, like literally. You're going to have the opportunity to use the chat box as well as the Q&A box. And we use those two things very differently, and you'll see them in your menu. The chat box and the Q&A box are two separate things. The chat box you're going to use as a way to communicate back and forth with the presenter. So the presenter might ask questions for you to respond to, or might ask what you think. You're going to use the chat box for that. The Q&A box is when you have a question for the presenter, because she might not be able to get to your question right away. And it basically stores the questions so that when we get to them, they'll be there ready to go. And I just want to remind you, all questions are good questions. So, you know, if for some reason you're not comfortable with the question, there is the opportunity to ask your question anonymously, but you also can put your name on it too. So Q&A box is for the questions. We ask that you just be courteous to one another, respectful, don't create any distractions. So none of you are on video. Really, the only way you can create distractions is if you use the chat box inappropriately. And so we like the chat box just to remain on topic, really focus on just what is happening with what the presenter is talking about. So we ask to stay engaged, participate fully, because the more you participate, the more fun you'll have, I guarantee it. So before we get to today's presentation, I just want to let you know that there are some new programs that are coming up in the summer, already thinking about summer. And the cool thing is some of them are in person, which I'm so excited about. So right now they're just save the dates because we're still waiting for approval to actually do them, but we're thinking we are going to get the approval to move ahead. So the first one is the Natural Resource Management Academy. So it's kind of like teen science cafes, just they're outside, and it's focused specifically on natural resources. So it's going to be one day, a Saturday, July 17th, and we hold it at the Green Mountain Conservation, the Green Mountain Conservation Camp, which is in Woodbury. And we're going to have 4 different workshops that you rotate through, as well as a couple of activities that everyone's going to do together. We're going to feed you lunch. We're going to feed you dinner. It's going to be awesome. And so I hope some of you will join us. The other opportunity that we have, this one is definitely happening because it's a virtual program. There's a program called Citizenship Washington Focus. Normally we send teens down to Washington, D.C., but we can't this year. So the program is being done virtually. And if you're interested in gaining skills and building on your leadership and citizenship, you might want to think about applying for this program. We're offering 8 scholarships, full scholarships. The registration is like $250. We're going to pay for it if you're a Vermont teen. And you can then participate in that program. You can go online to find more information about it. The deadline to apply has just been extended until May 15th. So you might want to think about that. And then the one I'm most excited about, we are hoping to do our Summer of Science, which is really the teen science cafes, just outside and in person. So I'm working with a bunch of people right now who are proposing their topics and trying to set dates and locations. But I can tell you, we've got one on where you're going to build your own rocket. We have one on geology. Not today's presenter, but we do have one on herpetology. We have one on microbiology and virology. We have one on, I'm already forgetting. There's some good ones. So hopefully this is all going to come together and a full announcement will come out next week. But stay tuned because I think some of you might want to participate in these because I think they're going to be good. All right, so let's get to today's presentation. We are going to be talking about Vermont's Herpetofauna, and our presenter is Dr. Brittany Mosher. And let me just tell you a little bit about her. So Dr. Mosher is an assistant professor in the Rubenstein School of Environment and Natural Resources at the University of Vermont. She grew up in the Hudson Valley of New York on a family vegetable farm, and she received her BS in Natural Resources from Cornell University. She got a master's degree in Fish and Wildlife Management from Montana State University, and then she got a Ph.D. in Fish, Wildlife, and Conservation Biology from Colorado State University. Her research focuses on understanding how diseases affect amphibian populations and how we can work to conserve them. So let's all welcome Dr. Mosher. Thank you for joining us today.
Thank you, Lauren. Thanks for the introduction. Hello to everyone who's checking in the chat. It's great to be here. I'm excited to get to chat with you today about Vermont's herpetofauna. And if the word herpetofauna is a new word for you, that's okay, because we're going to talk about exactly what that is. So let me just get my chat visible so that I can see any questions or comments or thoughts that you might have during the process. And I'm not sure that I can see my chat, Lauren.
That's okay. I can tell you how to see it on one screen if you'd like, or I can.
Yes, please.
Okay. So unshare your screen for a second.
Okay.
And when you go back in, don't go in present mode yet. Go in just as your PowerPoint. Before you go to present mode, open up the chat box.
Okay.
And then you should be able to move it and see it while you're presenting.
Oh, I see. Pop out.
Yeah.
Okay. Thank you very much. I definitely want to be able to chat with you all here today. Okay, it vanished on me, so you might have to moderate it, Lauren. I popped it up. Okay, thanks all for the warm welcome. I saw some welcomes in the chat before it vanished. I appreciate that. Today I'm going to talk to you about Vermont's Herpetofauna, but first I wanted to tell you a little bit about what it is that I do. So I classify myself as what's known as a population ecologist. And a population ecologist asks questions, in my case, about wildlife populations, including questions like where? Where does this species occur? And so on my little diagram here, maybe the species occurs in these orange areas and it doesn't occur in these white circles. I ask why. I try to figure out why some species are in some places and not in others. I also ask the question, how many? So you might imagine for something like an endangered species, we really care about how many of them are left and if we can do anything to make that number increase. So how many is important. And then probably the most important question of all is why? Why are species in some places and not in others? And why are there only a certain number of species? What things are making those populations grow and shrink? I use some fancy models. So this is just my plug for paying attention in math class. We won't talk too much about math today, but the way that I answer all those interesting questions is to collect data and to use models to figure out whether my hypotheses are right or wrong. And those are the things that help me figure out what factors might be stressing our populations of amphibians. This happens to be a wood frog. We're going to talk about wood frogs today a little bit. They're an amazing species. They have all these different life stages going from an egg to a tadpole to something in between a tadpole and a frog, right? It has legs, but it also has a tail. And then finally growing into the adult frog form that you might know. And then as Lauren mentioned in my introduction, I'm also a disease ecologist. And so the populations that I study are often being stressed by diseases. And this is just a picture of a toad. And I've put on some little clip arts here showing what a pathogen, if we could zoom way in, what it might look like on the toad's skin. And a picture here of me in the field trying to sample for that pathogen that causes disease. We're going to talk about that today too. So first question for you all is, we're here to talk about Vermont's herpetofauna. I am a herpetologist among these other things I've told you that I am. And I'm curious, what do you think a herpetologist is? And I think I've found my chat.
Nice. So question is, what do you think a herpetologist is? Put your guess in the chat box. KL says one who studies reptiles.
You're very close, KL. Anyone else have an idea?
So Amelia says someone who studies amphibians and reptiles. Lily says someone who researches and studies reptiles and amphibians.
Excellent answers. You are all on the absolute right track. A herpetologist is someone who studies or researches amphibians and reptiles. And so maybe another great question for me to ask you is, what are amphibians? When I say amphibian or I say reptiles, what kinds of species are we talking about here? What kinds of species fall into that category of amphibian or reptile?
Sage says snakes. Alice says frogs. So does
Okay,
Amelia.
we've got snakes, we've got frogs. Can you think of any other amphibians or reptiles? Oh, we've got turtles, excellent. Salamanders, wonderful. And Lily is making an interesting point here that I want to carry through to the next slide. So reptiles have scales, right? And amphibians, if you ever have held an amphibian like a frog, it has a really smooth kind of almost moist skin, right? So if you had sitting in front of us a snake and a frog, you might ask the question, why are we lumping these things together? Are they even that similar? Are there any good reasons to create this thing called herpetology that includes amphibians and reptiles? Why don't we just treat them separately? So I'm curious why you think we group them together as herpetofauna. And I'm already seeing great answers in the chat. Fuzzy is saying they're cold-blooded. KL is saying maybe they have some common habitat that they use. And those are really good suggestions. Sometimes you can just go back historically, and that might be a reason why we do something that we do. And here's a quote that I found. Early scientists called amphibians and reptiles foul and loathsome. The word herpes, which is in Herpetofauna, means a creeping thing. So early scientists thought they were a low-level life form, creepy, foul, and loathsome. And that's probably why we group them together today. Although I think that's a pretty crummy reason, and I think they are not foul, loathsome, or creepy. There are some reasons why it might make sense to lump them together, and those have been coming up in the chat. Ectothermy, which is the fancy science term for being cold-blooded, is one reason. Another reason is, of all the sort of vertebrate species, so species with backbones, they're probably the groups that we know the least about, right? We know a lot more about mammals and a lot more about birds, and to some extent, a lot more about fish. They're highly threatened, and maybe most importantly, they are completely awesome. So I just want to show you a little bit more information on each of these points that why it might make sense to lump them together. The first is that reptiles and amphibians are both ectotherms. And I could have asked this question in the chat, but an ectotherm is an animal that relies on an external heat source to regulate their body temperature. So 2 questions here. What kind of external heat source do you think they're using? And then the second question will be, what's the opposite of an ectotherm? Good. Yep, the sun. The sun is the external heat source that these ectotherms are relying on to keep their body temperature within a range that keeps them healthy. And great work, Gabby. So the opposite of a cold-blooded animal is a warm-blooded animal. We call them endotherms too. And so humans, we're endotherms. We have these fancy bodies that can allow us to adjust our body temperature. If I go outside, I don't all of a sudden become the same temperature as the air. My body can maintain a constant temperature. But amphibians and reptiles, they're getting that energy from the sun. And Wisteria's comment on rocks, totally true. That's why you see amphibians and reptiles sometimes basking on rocks, things that hold the heat. So I just wanted to show a quick video here. This is of a shovel-snouted lizard, which is a native species in Angola and Namibia. So the west coast of South Africa. And I want to show you what this species does. So it's having itself a little snack. What's it doing with its legs? Or more importantly, why do you think it's doing that with its legs? What's it doing there? What is this dance? Great work in the chat, by the way. Yeah, so they're doing this dance because the sand is hot. And essentially when some of their feet get so hot they can't take it anymore, they lift them up to let them cool down. Well, of course, when they do that, their other feet start getting really hot. So then they switch again, And the video goes on to show that when it just gets too hot and you can't switch your feet fast enough to stay cool, this species will actually bury themselves deep under the sand until the temperature comes down and they can come out and sort of be safe and operational. So starting at 120 degrees on the surface of the sand, as the shovel-nosed lizard use that shovel nose to dig down deep, their temperature drops. And that's what they need to sort of keep them going. So as we talk today, we'll just think about all the different things that amphibians and reptiles might do because they're ectotherms. They might have to be really conscious of how they operate. Okay, so I said the second thing, which was a good reason why we might lump them together, is because we don't know very much about them. So these graphs, the take home here is this gray area. So this is a pie chart. It shows of all the amphibian species on the left and all the reptile species on the right, what proportion of species are threatened. That would be in these warm colors, or whose populations are stable. That would be in these blue colors. And then the gray, which is what we're focused on, is what proportion do we not even know enough about to be able to tell whether the population is threatened or whether it's safe. So you can see it's almost a quarter of amphibians we don't know anything about. And a little less than that, but still a very large chunk of reptiles we don't know anything about. So we call this being data deficient, meaning we don't have data on these species. So that's another commonality between amphibians and reptiles. They're both vulnerable to a lot of threats. In fact, 43% of amphibians are threatened by some of these big drivers of population loss, like invasive species, climate change, habitat loss, disease, pollution, and overexploitation. And overexploitation means overuse by humans. So whether it's collecting these animals in the wild and selling them as pets, or whether it's in some culture, some of these species are eaten, it's overusing these species. And then my final point was, sure, let's lump amphibians and reptiles together because they are awesome. They're just amazing in terms of how they're shaped, how they're colored, the things that they've been adapted to do, and what their ecology is. So we're going to talk about a little bit of each one of these, and I hope that by the end of today, if you don't already agree, you'll agree that this is a really special group of wildlife. Okay, so here's my next question for you. We're going to talk about amphibians, and then we're going to talk about reptiles. For amphibians, there are actually 3 subgroups, and a couple of the subgroups came up in the chat earlier, but I want to know what you think those 3 subgroups are. What are the groups that make up amphibians? I could scroll up in the chat. Okay, we've got frogs, toads, salamanders. We got salamanders, turtles, and frogs. Those are very good guesses. Turtles. Okay, we're going to get this all sorted out today. There's a little bit of confusion here. The first group is frogs and toads. They're actually lumped together. Technically, they're all frogs. Toads are just a special kind of frog. So that's a little bit of a trick, but frogs and toads is one group. Salamanders is one group. And then we have this third group called the Sicilians. And so we're going to learn a little bit about these different groups here. Yeah, those who are saying turtles, that was a good guess. Hold on to turtles for when I ask this question for reptiles.
Brittany,
So, yes.
There's a question in the Q&A that I would, if you could answer quickly, it's just a definition. You use the word morphological. Could you just tell everyone what morphological means?
Yes, great question. I said amphibians are amazing morphologically, and morphology has to do with an animal's shape. So you can see here, this animal has these little dots on it. These seem to look like leaves. They have little spikes and little horns on their eyes. So their morphology has to do with the general shape of their bodies. Thanks for asking that question. All right, so we have these 3 groups. We've got the frogs and toads, we've got the salamanders, and then we have this wacky Sicilian, which I'll tell you more about what Sicilians are momentarily. We're going to start with the order Anura. This is the fancy science name for the frogs and toads. And Anura means without a tail. And so most of our frogs, they don't have tails. They actually do have a tail when they're tadpoles, right? But in their grown-up phase, we call them anurans because they don't have a tail. And if you looked at a pie chart of all of the amphibians, you'd see that the vast majority of them are the frogs and toads. We have over 7,000 species of them worldwide. 88% is the percent of amphibian species that are frogs and toads. This is a picture of a glass frog here, and it's called a glass frog because we can see right inside of it to its internal organs. So amazing frogs and toads. Then we go to the order Urodela, and Urodela, again, is a fancy science word for the salamanders. And just like we had the frogs and toads, and they're all technically frogs, we have the salamanders and newts. I haven't seen anyone say newt in the chat yet, but we do have newts, and they're kind of a special type of salamander. And I just love this quote by Bill Bryson, who's an author, who says, "Salamanders are interesting, and don't let anyone tell you otherwise." I agree with that. So if we looked globally, the whole world, and looked at all the amphibians, only 10% would be salamanders. Right here in North America, where all of us are tuning in from, 50% of our amphibians are salamanders. So North America is what's known as a salamander hotspot. And getting a little more specific, the Appalachian Mountains, which kind of go from the southeast up to pretty close to Vermont, but not quite, that is the true salamander hotspot. In the Appalachian Mountains, 60 or 70% of the amphibians that you have there are salamanders. So North America is a big deal when it comes to salamanders. You may not have known that you live in a salamander hotspot, but we have 9 out of the 10 major groups of salamanders that exist in the whole world have a member who exists in North America. And they can look wildly different, like these 2 pictures here illustrate. Okay, and then finally, the order Gymno, the Sicilians. This is this strange group that most folks don't know about. Sicilians means the blind ones. And Gymno, the Latin science name here, means naked snake. They're long and they're legless. They look a little bit like a snake, right? But you can kind of see on this picture on the right, they don't have scales. They have that amphibian moist skin. And a lot is unknown about these species because in part, they spend most of their life underground or underwater, and we just can't find them. But now you know, when someone says amphibians, you say, oh, did you mean frogs, toads, salamanders, newts, and Sicilians? And they'll be a little taken aback because this is the one people don't know. All right, so I think, Lauren, we're ready for the poll question, which is, which of these amphibian orders do we have in Vermont? So we've got Anurans, which is the frogs and toads only. We've got the Urodelans, which are the salamanders and newts only. Both Anurans and Urodelans, that would be frogs, toads, salamanders. Or do we have all of them, including the Sicilians, the blind? Go ahead.
I'm sorry, I launched the poll, so hopefully you all see it. If for some reason you don't see the poll, just put A, B, C, or D in the chat. But it looks like most of you are thinking through and starting to make your guesses in the poll.
I see the results coming in right now.
Yes. We'll give it a few more seconds. I know some of you haven't weighed in yet. Nobody will know. Just make a guess.
No one will know. Completely
no harm
anonymous.
in making an educated guess. All right, we're going to close the poll in 5 seconds. Take your opportunity to make a guess. Okay, so Jack didn't agree with Simon's answer. Okay. All right, I'm going to close the poll, and it looks pretty, a slim margin said D.
D. Okay, so in Vermont, we have both Anurans and Urodelans, but no Sicilians. So the Sicilians we do have in North America, but they're mainly in the South, in Texas, Louisiana, kind of right along the border with Mexico. So we're going to talk about Vermont amphibians today, but we're not going to talk about Sicilians because we don't have any. All right, I have another poll question, and that is, how many Vermont amphibian species do you think there are? 1 to 5, 5 to 9, 10 to 15, 16 to 20, or more than 20. So how many unique types of frogs, toads, salamanders, and newts do you think that we have in the state of Vermont?
All right, the poll has been launched. And again, if for some reason you don't see it, just put your answer in the chat. But it looks like you all are making your guesses fast and furious. We'll keep it open for another 5 seconds, see if we can get a few more responses. We have most people saying more than 20 and a split vote on 10 to 15 and 16 to 20. If one more person votes, maybe we can. But it does seem like the majority are saying E.
Okay, great. The majority is correct in this case. It's kind of neck and neck there. We have 21 species here in the state of Vermont. So we don't have Sicilians, but I still wanted to give you a little bit of info on Sicilians because they're really cool. They have elaborate reproductive specializations, meaning the things that they do when they have babies are really creative. Some of them, like this one on the left, they lay their eggs and then they guard the eggs. So they don't leave the eggs alone, say, good luck. They stick around and they protect the eggs from potential predators. And then there's another species that after the eggs hatch, the young that you can see here, the young have evolved to eat the skin of the adults. Right after the eggs hatch, the mothers, their skin goes through this transformation that creates all these extra nutrients. And then the babies are born with these special little teeth that allow them to pick off bits of the skin of the mother to give them a good early start on life. So I just thought those things were pretty neat. Sicilians are wild. We don't have any in Vermont, but it would be pretty neat if we did. And I see the question from Sage. It does not kill the mother. The mother can handle losing a little bit of skin, and it's no problem. How big are the biggest Sicilians? Some are as small as earthworms, but others can be up over a foot long. Good questions. So here are some pictures of our Vermont anurans. So we're starting with the frogs and toads. And you might know some of these species, but I just want to name them for you. We've got up on the top left and going across, we've got the American bullfrog, the green frog. Of course, this one's a little bit brown instead of green. The mink frog, the wood frog, the American toad, the Fowler's toad, the leopard frog, named for its spots, the pickerel frog, the gray tree frog, the boreal chorus frog, and the spring peeper, which you might be familiar with hearing by sound. I put a star on this boreal chorus frog because it's a species that we think is extinct in the state. It hasn't been seen since 1995, but there's still some hope that it does still exist. So amphibians are important in part because they're a huge amount of biomass in water. And biomass is essentially the weight, it's mass, right? It's if you took all the amphibians and you put them all in a bowl and you put them on a scale, that would be their biomass. So if you look across a habitat, in this case, a water habitat, you would find that amphibians are a huge contribution by weight. And that's a way we think about things because amphibians are out there, they're eating, they're excreting, they're being eaten, etc. And so from one site in Georgia, just to illustrate this point, they counted over 360,000 amphibian metamorphs, which is the stage right as you come out of water. So just as you go from tadpole to froglet. And if you weighed all of those, that would give us 1,400 kilograms of biomass. So amphibians are a huge part of biomass, especially in water where the tadpoles accumulate in large quantities. And Theodore's question in terms of what species eat Sicilians, it's largely, since many of them are quite small, it's mammals, things like rodents, and especially things that dig, because again, most of these are spending time underground. What do you think amphibians do in wintertime? How do they make it through our cold Vermont winters? We had snow here just, what, a week ago, a week and a half ago? What do? Yeah, they hibernate. It's not quite the same as what mammals do, but it's pretty similar. And actually, they have two different unique strategies that I want to talk about. Some of them, as Amelia mentioned and Lily, they bury themselves deep in the mud, maybe at the bottom of a pond or maybe on land. This is a picture of an American toad. You can sort of see it. What a nice job the toad has done of burying themselves. And some of these species that bury themselves, they can't freeze. So they need to find a really good spot where it might get very, very cold, but where they won't quite freeze solid. And so deep underground, under the frost line is one option. Near some water that flows is another option. Deep in the mud of a pond is another option. However, there are some species, and we have a couple of these in Vermont, that can freeze solid. So here is our little frog sickle. This is a wood frog. Wood frogs, they don't look for a place to hide. Maybe they go under a leaf or something, but they just wait for winter to come and they freeze solid. The way it happens is sort of magical, honestly. So here's a graph. It has some pretty fancy words on it, but the general idea is that when the temperature here on the y-axis is high, they're moving around, they're doing their thing, right? It's not cold out. But as the temperature drops, something called glycogenolysis begins. And basically, sugar accumulates in their body and starts protecting their organs. Eventually, as it gets cold enough, their heartbeat truly stops. They don't breathe and they're frozen. They don't move. And then as the temperature increases again, their heart starts beating, their breathing resumes, and they start to move. This is why wood frogs are the very first breeders we have in the state of Vermont, because they don't need to wait until the ice melts down underground. As soon as the surface melts, they come back to their state of animation, and they can look for a partner and start to breed. So those are our anurans, our frogs and toads. Next, I wanted to talk about our urodellans. These are our salamanders. And I've shown them here. We've got some neat species. This is the blue spotted salamander. This is the eastern newt. Sometimes we call it the red eft. This is the orange phase. This is called a red-backed salamander, a four-toed salamander, a Jefferson salamander. This one's got a funny name. This is the mud puppy. This is the spotted salamander here on the right. And then these three are found in streams. This is the dusky salamander, the two-lined salamander. You see it's kind of got two lines on its sides, and the spring salamander. So those are our salamanders. One of them is a newt. And on land, amphibians are also a big part of biomass. The red-backed salamander, in fact, has this special title. A study in New Hampshire many years ago found that if you were to pick up all these red-backed salamanders, and they are little, they're maybe the size of my pinky, that would be a full-sized one. If you were to collect them all and to put them on a scale, they would weigh more than birds weighed during the breeding season, which is when we have a lot of birds in Vermont. And their weight was almost the same as if you caught all of the small mammals, like shrews and mice and voles. So these are an incredibly abundant species. We don't see them very much because they spend part of their life underground. But just because we couldn't find them doesn't mean that they weren't there. And Gabby, you're right that mass and weight are different, but they do have some similarities that I think are useful for thinking about biomass. This is a group of salamanders that I wanted to tell you about today. We call them the Jeff complex, okay? So these salamanders are pretty amazing. And my first question for you, before I tell you how amazing they are, is how many sets of chromosomes do you have? And then as a follow-up, how many sets of chromosomes do you think these salamanders have? So let's start with the first question. How many sets of chromosomes do you have? Does anyone know? Good, you have 26 chromosomes total. And then for each chromosome, I guess how many, for chromosome 1, how many copies are there?
Brittany,
This is,
can you
yep.
just for clarification for Gabby, because she is a little young, what is a chromosome?
Sure, yeah, chromosomes are your genetic code. And so you have your own unique set of genetic code. And these things appear in chromosomes. And in humans, I'm going to give you the answer. Humans have chromosomes that are made up of genes and DNA. And we have 2 sets, one set from mom, one set from dad. Okay, so in the process of creating new life, genes from mom and dad come together, and the offspring has half from mom and dad, half from mom, half from dad. One set of chromosomes from mom, one set from dad. These salamanders are wild because some of them, here are sets of DNA here, some of them are what we call diploid, di meaning 2. They have 2 sets of chromosomes. That's like us. Some of them are triploid. They have 3 sets of chromosomes. Some of them have 4 sets, and some of them have 5 sets. So within this species group, individuals have all different amounts of DNA is what it amounts to. Humans, we all have the same amount of DNA. Our actual DNA, and whether it codes for brown hair or blue eyes or tall or short, that's all different. But in terms of the amount of DNA in our bodies, it's all the same. For salamanders, it can be wildly different. And even more interesting is that if one salamander had these 6 offspring, they could all have different numbers of chromosomes too. So some of the babies might have 3 sets, others might have 4 sets. So this complex, we call it the Jeff complex, because it's a group of salamanders. It's the Jefferson salamander on top, it's the blue spotted salamander here in the middle, and they can produce hybrids like this one on the bottom. Can you kind of see how this one on the bottom looks a little bit like this one on top and a little bit like the one with all the beautiful blue spots, but it's kind of in the middle? It has a few blue spots. It's much bigger, kind of has that gray background of the top one. So essentially, I just thought it was neat. These salamanders can take in genes and can decide how many to give to each of their offspring. And they could even choose to not give the offspring any genes except for their own. So they could clone themselves. That's called hybridogenesis. Or they can steal chromosomes. I call it stealing, but they're not exactly stealing from folks they mated with, and that's called kleptogenesis. So it's kind of a pretty interesting complex here that creates these hybrids like the one on the bottom, and all the hybrids are female. In case you're really excited about finding amphibians, I wanted to talk a little bit about how you would find them. One great place to look is in water, right? And you might, in water, find tadpoles. You might find adults. Sometimes you walk along a pond and you hear a little plop and someone's jumping in right in front of you. So if you had a net and you were really stealthy, you could capture tadpoles or adults. And you can also look for evidence that they've been there by looking for eggs. So these are the egg masses of 2 different species in Vermont, the wood frog and the spotted salamander. And if you get really good at your egg masses, then you can figure out who's been to that pond. But you can also find them on land. As I mentioned before, this is our redback salamander. This is the one that's super common, right? Super common, lives a lot of its life underground. And so we key into the idea that amphibians have this moist skin when we think about how to find them. They need to keep that skin moist. So most of the time when you find them, it's when they're under a rock or a log, in contact with the ground, right? So when I go out in the woods, sometimes I flip small logs and I look for salamanders. And this is a species you can find in a lot of places along the East Coast. And then the final way to find amphibians would be on what we call big nights. And big nights are these predictable spring evenings when amphibians migrate from the woods to their breeding ponds. So they don't live at the wetland all year. They live in the forest during the summer. They overwinter in the forest for the most part, and then they have to get back to the pond in the spring to breed. So on rainy nights, and actually in Vermont, it's a rainy night, and it has to be at least a little bit warm, so above freezing, above about 40 degrees, you might find amphibians crossing roads or walking through the forest toward their breeding ponds. So that's another time when a lot of folks get out to look for amphibians. And you can actually help them cross the road. And Gabby, you're right. There are some frog species that live in water pretty much all year. There are definitely species that hibernate buried in the mud at the bottom of a pond or stream. Oftentimes they'll breed somewhere and then move to a different kind of water to spend the rest of their life. But you're right, not all of them live in the forest. And this is just my note that if this sounds exciting to you, make sure you bring an adult and make sure you're very safe. This is a picture of one of my students named Izzy, and Izzy is demonstrating excellent safety. She's got a headlamp on. She's got this nice fluorescent vest. So if she's helping salamanders cross the road, she is seen by any oncoming traffic. And when you handle amphibians, you want to make sure that your hands are a little bit damp because they're very prone to drying out. And you don't want to have any sunscreen or lotion or bug spray or anything like that on those hands. And you want to hold them gently, no squeezing. And then you want to release them as soon as possible. You know, maybe you're helping them cross the road and you need to do that, or maybe you just want to take a quick look, but it does cause some stress when they're being handled. So after you've had your look, you know, release them right back where you found them. So amphibians are in decline. We mentioned this briefly at the beginning, and if we look back historically between the year 1600 and 1970, there were only 2 species that we knew of that went extinct. Well, by 2004, almost 450 species are critically endangered. So it does seem like things are getting worse for amphibians, that they are increasingly threatened. And they're threatened by a bunch of different things, including land use change, pollution, introduced species, and so on and so forth. I want to talk about a couple of these today. The first is habitat fragmentation, and we're thinking about roads and things like that, housing developments that separate habitats. And just as I talked about on the other slide, some of these amphibians, they need both the woods and the wetlands, right? So if we put something in between those two places, it starts to get really hard for them to use both habitats that they need for their life cycles. So that's fragmentation. Another one that's a threat is disease. And I want to talk about 2 fungal pathogens today, so 2 fungi. The first one we call BD, and the second one we call B-Sal. It's just an abbreviation of their super long names here. And this is how it works. BD and B-Sal are these little tiny spores, right? They're from a fungus, like a mushroom. They have tails. The tails allow them to move through water. And if they encounter an amphibian, they can bury themselves in the amphibian's skin. The amphibian goes about its daily business, and those spores are continually burying into the skin, and they're creating these larger sort of cysts that will ultimately rupture and spill spores back onto the amphibian skin and into the water. BD has been around for a while, and we think that it has caused declines in 500 species. B-Sal, we just learned about fairly recently, and so far we don't think that it's in the US yet, but we're very worried about it spreading here because we are that salamander hotspot, right? If B-Sal gets here and it affects salamanders, then we might lose a lot of our salamander species. So here you can see are some little lesions that are formed when the spores bury into the skin. We look for BD and B-Sal by rubbing a little Q-tip on the skin of amphibians and then sending that Q-tip to a lab where they look for the DNA of BD or B-Sal, the fungus. It looks for the spores. If we find evidence of the spores, then we say, yep, that little toad was infected. The things you can do to help are to never release a pet. If you have a pet amphibian or a pet reptile, never release it into the wild. You can talk to your friends and family about the same, asking them to never release pets. If you go out a lot to a bunch of different wetlands in rapid succession, you could think about rubbing down your boots with a little bit of bleach, making sure they get nice and clean before you move from site to site. And you could keep an eye out for strange lesions, so those strange little bumps on salamanders, and you can report it to someone like me if you ever saw something suspicious. All right, now we're on to reptiles. So that was our talk on amphibians, but we've got to get to the other half of the Herpetofauna. The first, we're going to talk about reptiles, and there are 5 groups here. We have the alligators and crocodiles, the snakes and lizards. Sorry, I guess 4 groups here. The turtles and the tuataras. This one down in the bottom right corner, it looks like a lizard, but it's actually a tuatara, which is something that's a little bit different from a lizard and gets put in its own special group. There are only 2 species of tuataras in the world, and they do not exist in North America. All right, so here's a poll question for you all. We're moving on to reptiles. How many Vermont reptile species do you think there are? So you have to kind of assess in your head, all right, do we have crocodiles? Do we have tuataras? Do we have lizards? Do we have snakes? Do we have turtles? How many species? Go ahead.
The poll's been launched. Give me your best guess. It looks like very even on 3 of the categories, with a favored being 10 to 15 species.
Okay, awesome. There are 19 species. 19 species in Vermont. 12 of them are snakes and lizards. Here they are. We've got the garter snake, the water snake, the Decay's brown snake, the ring-necked snake, which has this nice ring around its neck, the milk snake, the rat snake, the ribbon snake, the timber rattlesnake, the racer, the red-bellied snake, the green snake, and the common 5-lined skink. So of our 12 species of squamates, this is the group that includes snakes and lizards, 11 of them are snakes, and we have a lizard. This is something that surprises a lot of Vermonters. What? We have a lizard? We do. It's the common 5-lined skink. Even though it's called the common 5-lined skink, it's not very common. It's actually endangered in the state of Vermont, and they're found only in Rutland County. And just a question in the chat, axolotls are amphibians, not reptiles, and we don't have them in the state of Vermont.
And I'll just jump in for a sec. Gabby asked in the Q&A, is a skink a lizard?
It is, yep, a skink is a lizard. A skink is kind of a special name, a subgroup of lizards. So lizard is the general term and a skink is the specific term. And you see these pictures look pretty different. The young skinks have this bright blue tail, but as they age, if they're a male that's ready to breed, they get this red coloration and they lose a bit of the blue on their tail. So this is a species you may see in the state of Vermont, depending where you live or where you go, but we're pretty proud to have a lizard. I wanted to ask, what's the largest snake we have? The largest is the eastern rat snake. This is a picture of me holding one that's maybe 3.5 feet long, but they can get up over 6 feet long. This is just a list of all of our snake species in order by length. And here's the eastern rat snake all the way to the right. The largest one that was ever caught was 75 inches long. So quite long indeed. On the other end of the spectrum, the smallest is the red-bellied snake, and they max out at about a foot long. And you can see up here in the top right, when they're born, they're super tiny. You can see them cupped in this person's hand. So we've got a big snake, we've got a small snake, and we've got all these snakes in between, which is pretty cool. I want to ask you, of those snake species, especially, I guess, if you are a Vermonter and you might have heard of some of these species before, which species do you think is the rarest? What do you think is our rarest snake species in the state? Go ahead and just post in the chat if you have a guess. Probably not the garter snake, right? Does anyone have a guess of what snake species might be rare here? We've got a guess for rattlesnake. We've got a guess for water snake. Yuri, you're correct. The timber rattlesnake is our rarest species. It is also only found in Rutland County, so it's not something you're very likely to encounter. Rattlesnakes and skinks actually live in the same area. They're a really shy and timid snake. So if you walked by one on a trail, you probably wouldn't have even noticed. It's our only species that's venomous, meaning that a bite can introduce a toxin to your body. And in the state of Vermont, they're endangered. And there's a lot of effort going into trying to conserve them and keep them around. So consider yourself very lucky if you ever get the chance to see a Vermont timber rattlesnake. There are not very many left. The most common, on the other hand, is the garter snake. This is a map of all the towns in Vermont where garter snakes have been seen. And as you can see, they've been found in every single town. Now we're going to move into turtles. The fancy science name for the turtle group is the testudines. And we have 7 species here. We've got the painted turtle, the musk turtle, the map turtle, the wood turtle, the spotted turtle, the spiny soft shell turtle, and the snapping turtle. And you're right in the chat. It would not be very good luck if you were bitten by a rattlesnake. But the vast majority of people that are bitten by rattlesnakes are trying to handle them, which is something that you should not do with correct training. Yep, Gabby, tortoises too. We do not have any tortoises in Vermont, probably. We've had some box turtle sightings recently. Box turtles are technically tortoises, but we didn't think we had box turtles in Vermont. So right now we're trying to figure out, were these pets that someone released, or have there been box turtles here all along that we didn't know about? It's a pretty interesting question. So I've got a question for you about turtles. How do you think they breathe underwater when they're spending the winter there? So in like this time of year, they can come up to the surface, right? Get a breath of air. But what about in winter, where they're burying themselves in the mud for the most part, and there's ice, and they can't get to the surface? Okay, do they have gills? Maybe. Are they breathing through their skin? Kind of. Or their ears? Or through their shell? I don't know if anyone out there has seen Frozen, but there's actually a part where Olaf says, "Did you know turtles can breathe out their butts?" And Olaf was actually a little bit right on this one. So turtles and birds and amphibians and other reptiles, they all have this outlet, I'll call it, the multi-purpose hole for peeing, for pooping, and for mating. It's all one hole, and that's called the cloaca. Cloaca. And so turtles, when they're hibernating, they do this thing called cloacal respiration. And this should say pumping. They pump water in and out of their cloaca. And in the process of doing that, they can pull the oxygen out of the water. And that leads to headlines like this: "The secret to turtle hibernation, butt breathing." So that's just something you might not have known about turtles until today. I wanted to make a quick vote for what do you think the most colorful reptile is in the state of Vermont. I put up 3 good contenders here. We've got the 5-line skink that's got that beautiful blue tail. And remember, I showed you the other picture where the males have that bright red face. This is the ringneck snake, where the top is pretty drab, just gray, right? But they do this display when they're threatened, where they flip their body over. And you can see that their belly is red and yellow and orange. And then we've got the painted turtle, which has all these beautiful lines on it. They're green and lime colored and scarlet. Which of these in the chat, I'm just curious, is the most colorful to you? A lot of votes for the ringneck is what I'm seeing. Yeah, this is a pretty striking picture. The painted turtle, says Yuri. Yep, the painted turtle is an incredibly beautiful species. They're all winners in my book, but I just wanted to show you some of our most colorful species. Reptiles also have a lot of different threats, some of the same ones that amphibians have. Land use change, so land being converted from conserved land to housing developments or industry or roads. Pollution, climate change, emerging infectious diseases again, and again, this idea of exploitation and trade. So here are some pictures. This is a picture of a form of pollution, right? Plastic bags in the ocean. And marine sea turtles, which we don't have in Vermont since we don't have any ocean, they eat jellyfish. And they actually often have been found dead because they're consuming plastic bags that look an awful lot like jellyfish, right? When they're floating in the ocean. This is a species here called the earless monitor lizard. And this is a species that's in decline because it's being captured in the wild and sold as a pet. And then here we've got a snake that you can see, it's something a little funny going on at near its face, right? This is another skin fungus. It's different than the amphibian one, but we sample for it in the same way using those little Q-tips. And this is a new disease called snake fungal disease that we've found in Vermont and we're pretty worried about. So to conclude, I just wanted to tell you what I do for a job. I'm a professor at UVM, and that means part of my job is teaching. And I teach a few classes. One is herpetology. This is my herpetology class. We went out to a wetland, to a vernal pool, meaning a semi-permanent pool that usually dries up at some point during the year. We went to look for egg masses and to catch amphibians and to identify them. And I conduct research. So one of my students, this is Matt. Matt is studying how a wildlife underpass, so a culvert basically that runs under the road, how that is helping amphibians migrate. So here you can see Matt is in the underpass, and here at night on one of those big nights, we have salamanders circled, and they're using the underpass to get from one side of the road to the other. So hopefully that underpass is reducing the number of amphibians killed on the road, but that's what Matt's project will answer. This is Lindsay. She's studying this beautiful eastern newt species here and is trying to figure out where they're most likely to occur. You hear people say newts are everywhere, but they're not everywhere. So we're trying to figure out what factors influence whether newts do or do not occur in a particular place. And this is important because newts are one species we're really worried about with that B sal that I mentioned, that new pathogen. We think it could be really bad for newts, so it's a good idea to figure out where newts are before that pathogen arrives. And then a final project led by my student Reed. Reed is looking at how amphibians that come together, amphibians of different species that all appear in one wetland to breed or do something like that, how they might be altering disease dynamics for one another. You might be kind of thinking along these lines with COVID, right? We've heard the idea about super spreaders and super shedders. And with amphibians, the question is, are there some species that are driving up infection rates for other species? Are some species more susceptible than others? Who's who matters? And how does who is present in a wetland influence how the disease progresses through those animals there. So that's a snippet on my research. I hope you learned a little bit about Herpetofauna today. I'm excited to answer any questions that we haven't gotten to yet. And I wanted to give you this website here, the Vermont Herp Atlas. is a great resource for our Vermont species. It's got a ton of information. A lot of the information and the photos from today came from that site. And it's also a place where you can upload an observation. So if you find a species in a new place and you want to report it, that's the place to go. And they can add it to some of those maps that I showed. Thank you all for your great attention today. And please go ahead and put any questions in the Q&A.
Yeah, this was great. So while you guys think about your questions, you might want to put in the Q&A box. I'm going to launch one last poll. We always like to get some feedback. So a scale of 1 to 5, let us know how you thought today's cafe was. And then scale of 1 to 5, 5 obviously being the best. How much did you learn today? So go ahead, take that poll. And if you have any questions for Dr. Mosher, pop them into the Q&A box because she's here for a few more minutes and is happy to take questions. So we'll see what comes in, but just, let's see, I'm gonna keep the poll open just another probably 20 seconds, try to get the last few of you to respond.
Thanks for all your nice comments in the chat, folks. It
Yeah,
was great to have a group that participates so nicely. I hate
know,
being
right?
here feeling like I'm talking to myself, so it was nice to get to talk to you.
Exactly. All right, I'm going to end the poll right now, and I do see that we have a question in. Oh, it's just a thanks from Wisteria in the Q&A.
Thanks,
for
Wisteria.
putting that, Wisteria. There is no question. Okay, well, if there are no questions, I think this is a great resource that Dr. Mosher, I would hope you are going to go check out the Vermont Herp Atlas, and maybe you'll all be citizen scientists and start contributing. Sorry, so Amelia actually just popped a question in. What is your personal favorite species of Herpetofauna?
Oh, what a question. You know, as strange as it may sound, with all these wild and wonderful species we talked about today, the American toad is my all-time favorite. I think they're one of the very first species that I ever caught as a kid. And then as part of my research, when I was working on my PhD, I studied a toad species that lived in Colorado. And I just think they're such a neat species. They're beautiful. You can find them almost anywhere. So I'm going American toad. Thanks for the question. Nice.
Well, let's all thank Dr. Mosher for a fantastic and very colorful presentation with all those great photos. So hopefully you all, it is going to be rainy for several days now. So maybe you'll all get out there and do some searching around and see what you can find. So thanks for joining us today. And hopefully I'll see you back next week at our Teen Science Cafe next Wednesday. So everyone have a good rest of your day. Thank you.
Thank you all. Bye-bye.